First, I really like to thank you for the invitation and for the very kind introduction. And I also want to thank Kyla for helping steer my lab toward some really cool, interesting new directions. And hope I don't say too much of what she said, but I think we agreed we're going to be complementary rather than so overlapping. So I'm going to tell you today about two things, I guess. One is, the first thing is the title of the talk, which is kind of following up on what Kyla told you, with the idea of using iPSC models to track disease evolution and maybe test therapeutic strategies. And one cell at a time means we're we're kind of going towards single cell imaging, as Kyla showed. So I think I show this slide 3 times in every course I teach. The idea that disease progression starts a lot sooner than the time that the typical clinical intervention happens, right? By the time we get to the point where the patient sees a doctor, they already have significant disease burden, and basically clinical interventions often focus on symptom management. And I think one of the things that I've really been excited by in terms of iPSC derived models and organoid built models is that it allows you to kind of track this preclinical or subclinical progression in time and really get a handle on things that predict risk, that drive the early initiating events, that allow you to detect those events, and that ultimately, hopefully, will allow us to intervene clinically earlier, or at least differently, with an idea of building sort of a personal health plan. And I know this slide is focused on the genomic aspects, but I think that what we're interested in is actually tracking the disease evolution itself. So I really think it's a missing piece of the puzzle as a basic scientist trying to understand how disease evolves. So the disease that I'll focus on today is the one that Kyla talked to you about earlier. Dilated cardiomyopathies, they slot into a larger family of cardiomyopathies that includes hypertrophic variants of hypertrophic version of the disease. Some aspects of congenital heart disease overlap with these diseases, neuromuscular disorders, and channelopathies that lead to kind of 3 sort of phenotypes: the dilated cardiomyopathy phenotype, the hypertrophic cardiomyopathy phenotype, where you get wall thickening, and then the arrhythmogenic phenotype, which is more driven by changes in ion channels, right? So these are all structural changes in the genes that are important for cardiomyocyte function, and we know that they sort of overlap with respect to phenotype and disease evolution. They may start with one and end in another. Many of them are caused by genes, as we understand that, but they also have, in addition to all of these genetic causes, there's probably 30 different genes that have been linked to cardiomyopathies. But there's also a lot of evidence that certain viruses, and it probably included COVID-19, that can drive cardiomyopathies. There's certainly some role for systemic immune-mediated disease. There's an autoimmune component to some of this. There are exposures in our environment which may actually lead to the development of cardiomyopathy. And certainly there are some chemotherapies that most importantly, I suppose, as anthracyclines like doxorubicin are known to drive cardiomyopathies. And of course, there are particular cases of metabolic disorders and peripartum disorders that may lead to an idiopathic forms of the disease. A lot of the disease is not understood, but I think as more and more sequencing happens, there'll be fewer idiopathic cases. But the causes are diverse, for sure. And for a long time, my lab was sort of interested in the signaling in the cardiovascular system from the perspective of a G protein coupled receptor person. We were interested in what controls decisions regarding whether or not the heart gets bigger in a physiological or a pathophysiological way. So how it gets bigger, what determines the in the few responses that cardiac myocytes show in response to stress, beat faster, undergo hypertrophy, or die. What controls those decisions, And then there's this stage where heart failure patients go from compensated hypertrophy, where they're still managing to produce enough function to maintain activities, toward cardiomyopathy and ultimately heart failure. And the interesting thing is it's the same G protein coupled receptors that drive all of these things, right? So it's a wiring question. You have GQ coupled receptors like the alpha-1b or the angiotensin 1 or the endothelin receptors. You have GS coupled receptors like the beta adrenergic receptors, and you even have a role for some GI coupled receptors. And I don't name them because they're probably the same receptors that are coupled to multiple G proteins. But there is this cycle of the heart perceives reduced circulating volume and changes in blood pressure, and it activates neurohormonal loops that involve the sympathetic nervous system, the renin-angiotensin-aldosterone system, and the endothelin system, which then cause all of their effects that are designed to maintain pressure, but that also at the same time drives additional left ventricular systolic function that followed some initial myocardial injury, and then it's a cycle that gets that worsens with time, ultimately leading to heart failure. So it's the same receptors that drive all of these processes, and so we wanted to understand them in a patient-derived way. And Kyla showed this slide and said it that iPSCs and tracking cellular signaling, they're almost a match made in heaven, at least for a basic scientist. So we now can get patient-derived blood samples, turn them into iPSCs, and then turn them into different types of cells that are important in the myocardium. And then ultimately, we want to mix them together and make co-cultures and then three-dimensional organoids that would better reflect the actual changes in the organ itself. We're not quite there yet. We're still trying to work out the cardiomyocyte cultures themselves. But the idea is that over the years, we and others have developed tons of tools that allow us to kind of track those changes in a more relevant physiological model, assessing morphological, electrical changes, the genomics that we can do, and then the screens that we can do to assess whether people are at risk for different forms of cardiomyopathies later. Ultimately, all hopefully ending on improved clinical intervention, as Kyla said. So for me, this kind of started back in 2012 when Joe Wu published this paper. I mean, this paper was published only 7 years after Yamanaka showed how to make iPSCs out of adult human cells, right? Or even 6 years after that. But basically what Wu showed was that you could actually take patients who had a particular form of familial dilated cardiomyopathy, so changes in the troponin T gene, and he could grow them into cardiac myocytes from stem cells. And then what he showed was there was a phenotype that was organized or disorganized in terms of sarcomere organization, had hypersensitivity to adrenergic receptor stimulation, and showed big changes in contractile function and calcium handling. But he also showed in that model that you could treat those cells with a beta blocker and it improved their function. And it showed, kind of what was seen in clinical trials, right? So it gave us some hope that we could use these models to track disease evolution and test therapies. So we've been doing that ourselves now in collaboration with a couple of groups at the Glen, who I'll thank profusely a bit later. And we've been able to get a population of cardiomyopathy patients who have different causes, right? And the cool thing that we can do then is you can make them, and I really thought Kyla was going to show this, because it brings a tear to my eye every time I see it, when we can actually show that these cells beat in culture, right? They're really becoming cardiac myocytes. Once they're stem cells, it takes about 2 weeks to do that, and it's still rather miraculous to me. Yeah, so the idea is we could build a pipeline, and that would start with understanding some of the features that are important, that are relevant to each patient, genetic and genomic characterization, making them into cardiomyocytes or other cell types, understanding the electrical properties and the phenotypic properties, and then do what my lab has become good at doing, which is characterize cellular signaling events, which is what Kyla talked about, measuring biosensor profiles and combining that with microscopy-based measures of cell phenotype. And for me, this is the problem that we're after, right? So drug discovery in the old days was slow, involved an animal model or tissue from animals, but we had a really good success rate. Then we cloned everything and expressed things in cells that were not relevant, and built these beautiful high content, high throughput system, and we failed. And what I think stem cells from patients gives us back is this human relevance, right? So ideally, we'll not only be able to do drug discovery in more physiological and more pathophysiological relevant models, but even we'll be able to individualize that from a patient perspective. So Kyla talked a bit about, like, we have a toolbox of resonance energy transfer based sensors. A lot of them are based on FRET. A lot of them are intensiometric. The neat thing is that now we have versions of them that can be, I think Kyla also said that, they can be localized to different parts of the cell, to the cell surface, to the myofilament, to mitochondria, to the SR, to the cytosol, and even to the nucleus. So we can really kind of understand the location of where these signaling events are occurring in response to a given stimulus. And the other thing that Kyla showed you is that we can now do this one cell at a time, because, well, I won't say COVID was enjoyable, but COVID actually gave us time to think. And during our first time away from the bench, Jace Jones-Taba, who I'll thank more formally later, had time to develop and think about how to make our biosensors amenable to single cell analysis using high content imaging. And so getting away from bulk analysis and toward individual responders really kind of gives us a lot of power to capture phenotype one cell at a time. And I'm really happy we did that. Our initial ligand panels were the ones that Kyla described to you. So angiotensin receptor, or beta or alpha adrenergic receptor, or endothelin, focusing on the pathways or on the receptors that are both involved in the normal function of the heart and in the transition to heart failure. And I just want to show you this slide. So this is kind of a busy slide. Kyla went through them individually, but this is just 3 different subjects, okay? One is a control subject who's male. The other is a male patient with cardiomyopathy, and the other is a female patient with cardiomyopathy. And I don't want you to focus on any of the details. This is looking at ERK signaling using an ERK FRET-based biosensor. But what I want to show you is that, these patients look different from one another. And we've all come to realize that heart failure in women is really a different disease than heart failure in men. And I think this way to track how the drugs that we use, how the signaling systems that we think are altered in heart failure are different in between men and women. And so I think that all I want you to focus on here is that you can see that the profiles that you get for looking at one biosensor downstream of several different ligands is different when you compare a male patient and a female patient who suffer nominally from the same disease. So I think this power that the single cell approach gives us is really quite striking. The other thing that high content imaging gives us at the single cell level is the ability to kind of paint cells or to classify structural features morphologically as that, so we can see using high content imaging how different cells are changed. And I took this slide from another one of my students, Allison, who wants to do this. But the real power of high content imaging is being able to stratify differences between cells at the single level. And so combined with our imaging for signaling, we think we can get a lot of high content morphological information that can be combined with signaling information, with the ultimate goal of being able to cluster patients into groups, right? So maybe we can make predictions about which drugs, and I'll come back to this, might be better for an individual patient, or which combinations of drugs might be better. And you think we do that now, but we don't really, and I'll come back to this notion a bit later. So the other thing that we're really interested in understanding is the wiring, as I said. So we have biosensors that can track all downstream signaling from a given receptor, both horizontally into different pathways, and then down each pathway vertically, right? And so the idea is that we not only can track the activation of a G protein, but a lot of the signaling downstream. And we have FRET versions and bioluminescence resonance energy transfer versions. And the idea is to really plot efficacy down at the pathway level when you're looking at multiple pathways. It's no longer a two-dimensional dose-response curve. You get this kind of Stephen Hawking type cube that you need somebody smarter than me to analyze. So if I just give you one example of that, this is signaling. This is a paper in collaboration with Stefan Laporte's lab where we looked at signaling downstream of the angiotensin receptor. If you look at angiotensin, it activates all of those pathways. But you can also develop ligands or identify ligands that only activate a subpopulation of those pathways, this idea of being able to track bias signaling. And the other thing is, like, if you go down one arm of the pathway, so GQ, you can split it into GQ and what's downstream of GQ, and you can actually track things like integration of signals or where signals diverge in a meaningful way using these different biosensors that track both proximal signaling events and distal signaling events. So that's kind of where we're going. And if you think about how that might impact therapeutic decisions, the conventional sequencing of failure of drugs for heart failure is this. It's this for all patients, and it takes at least 6 months for these drugs to be onboarded individually, right? So what if you could find a way to kind of onboard them more quickly? And so John McMurray and Milton Packer have been thinking about this. Is that they were thinking about it in patients. Instead of up titration of 6 months, they would up titrate, different combinations much sooner. But we think, and in my discussions with John McMurray, he thinks we might be able to do this with stem cell derived cardiac myocytes from individual patients. So can we adapt the sequencing of drugs patient by patient rather than just using the conventional sequencing that is used for everyone? And then the other thing we want to do is that, can we use it to evaluate risks? So it's known that a subpopulation of breast cancer patients and other cancers who are treated with doxorubicin develop a cardiac myopathy. And some of that happens, while the patients are being treated with dox. Some of it happens within a year of being treated, and some of it happens 10 or 20 years after that. How do we stratify risk? How do we know whether or not we can use these drugs effectively, right? 40%, 8 to 10% will show doxorubicin-induced cardiomyopathy of women treated with doxorubicin for breast cancer. Among the survivors, 20% will get heart failure in the next 5 years, and 40%, that number increases if they're treated with other drugs like Herceptin, right? So it's something that we want to be able to model, and we think we can do that with our stem cell-derived cardiac myocytes too. So are these weasel words? Can we do precision medicine or personalized medicine? So we already know we can make different cell types from iPSCs. There are issues, as Kyla indicated, with maturation, but we do already know that they recapitulate phenotypes that are patient to individual, specific to individual patients, and also disease relevant, right? Which is important if we're going to do that. And they've already been used to model a lot of different cardiovascular diseases. And if you go beyond cardiovascular, there's a lot of neuronal diseases, et cetera, et cetera. And we think it would be a really interesting platform on which to build a drug discovery pipeline. And so, but we need to have, we need to be able to do this in an effective way. And we need to do it beyond single cells, right? We need to be able to build them into 3D models. So I think that the idea is that the most effective models would be to take patient-derived iPSCs and build three-dimensional organoids where all of the tools that I've talked about using and that Kyla talked about can still be used in these three-dimensional contexts, and then use that as our basis. So it's a scaling issue, right? The main reason we use two-dimensional cultures is they're easy to use. But in the heart, we have all kinds of things to think about with respect to how those cells are connected, not only to each other, but to the matrix on which they contract against, right? And how they're fed by blood vessels. So it's not an easy problem, but it's one that I think that is coming going forward. Okay, if you'll indulge me, I just wanted to spend the last few minutes talking about, they gave me the great honor of succeeding Michel Tremblay, and I don't say I can replace him. I only admit that I can succeed him. And when we started the MRM, we had 5 commitments, and some are pretty straightforward: to expand our footprint in the stem cell and regenerative medicine world, to kind of make contributions to understanding and translating the biology, to build and develop translational pipelines, importantly, to train high quality personnel with a focus on interdisciplinarity, and of course, to maintain a commitment to the ethical, legal, and social impact of our work. And if you think about it, as I've had to now because they gave me this job, it does align well with both the Faculty of Medicine and Health Science and the university's strategic plans, right? All of these diseases are healthcare across a lifespan, infection, inflammation, cancer, neuroscience. Our researchers are involved in that. Patient-centered continuum of care. I think that well, what I've been talking about are these two things, like precision approaches to personalized medicine and patient-centered medicine. And then understanding the potential of the human brain and nervous system. Well, I've talked a bit about heart, but there are many efforts at McGill to model disease in the nervous system, right? And then advanced biomedical and health sciences for healthy populations. I think that we fit really well into the university's strategic plans. So what am I wanting to do to keep the MRM moving forward? And I want to acknowledge my gratitude for Michelle for getting it moving forward in a really good way. So I want to build on our existing strengths. We already have more than 100 PIs and several hundred trainings, and that's a huge critical mass across the main campus and many of our affiliated research and clinical sites. We have the tools to build something big. I want to strengthen our education programs. We've already built a small education platform, a graduate certificate in stem cells and regenerative medicine that we're now in the process of converting to a thesis-based MSc program. We already support a lot of trainees to attend meetings, travel awards, virtually or in reality, right? And we were going to set up a better connection to the School of Biomedical Sciences and the School of Medicine, I think, and other schools in our faculty to kind of strengthen these educational programs. We want to kind of, by connecting the people at McGill and beyond, we want to build translational opportunities and programs, right? We already provide seed funding for interdisciplinary research. Things like this, the MRM Talks and our MRM Symposium, when it goes back, once we've dealt with COVID again, we're going to help connect us better using these venues. And I've tried to tell you about something I'm interested in personally, which is, strengthening the drug discovery and disease modeling components and returning a biological and hopefully a clinical context to drug discovery. So I think that means that, like, meaningfully as a director, I have to engage you to help us build and sustain research cores, to help us kind of put together and push for multi-investigator grants, to help us build and develop meaningful partnerships, internally, and beyond, and to help us move toward rare diseases. I think that's we can engage all sorts of resources that way. To build in education at all levels of our research, and to build in translational training and courses to create a shared language amongst our student body and beyond, right? Our students who are going into medicine, our students who are going to other healthcare professions, our students who are going to become scientists, they all need to be able to talk to one another. And I kind of view that as a mission to kind of give them a shared language. So, broadening connections with the medical school and other professional programs in the context of research translation, establishing connections beyond our faculty, in the Faculty of Sciences and the Faculty of Engineering, broaden our connections with Stem Cell Network, with TACEL and other universities to establish McGill and the MRM as a key node in these activities, right? And in their activities too. And of course, to create and strengthen partnerships with pharma, biotech, and I think instrument makers as well as a line of work. And then work with university advancement in the context of both the McGill and the Faculty of Medicine and Health Sciences strategic aims. So that's kind of what I want to do. This is what I see. And what I've seen for years is that we remain highly siloed. The MUHC and McGill University, to me, act entirely as separate entities and sometimes. But if you view them separately, the pieces at McGill are extraordinary. And if we can break those silos, we could really get McGill to live up to its reputation, right? I mean, it's earned reputation, but I want to maintain and grow that too. And I think the MRM, being so diversely placed at everywhere at McGill is uniquely placed to help, but we need to grow it. So that's what I want to do. All right, just to thank the people who actually did the work. I'm going to start, well, with Kyla, because Kyla is, every time I see that, that movie of cells beating, I tear up. I want to thank Jace because he helped us build that single cell pipeline that we're using. And then my Heart in a Dish team, Grace Mazura, Uman Sadigian, Kara Howie, Allison Jang, and Darlene Petren, and our newest member, Karima Alim. I also want to thank people beyond my lab, Jason Tanny, who has been a great collaborator, teaching me how to do genomics in the modern way. Nadia Gianetti and Natalie Gendron, who works in her lab. Renzo Cicceri and Ida Darish, Kashif Khan, and Jeremy Zwag, who work in his lab. This is the team that allows us to get patients who have cardiomyopathies, and this is the group that actually makes our iPSCs. And Bruce Allen and David Chatanet and Tom Durkin all helped at various stages of developing this. And then I want to thank the CIHR, Heart and Stroke for funding part of it, and from a very generous donation from the Courtois Foundation to set this up in my lab. And with that, I will thank you and again for inviting me, and I'm happy to take questions.